the cell, subcellular organelles, and cell membranes. So, what is a cell? Cell is a structural and functional unit of life that is capable of performing all the essential life processes such as growth, metabolism, reproduction. It is the smallest unit of an organism that can exist independently or as a part of a multicellular organism. Now, there is a little activity for you. Take a piece of paper and identify which organelles are present, which are the organelles which are labeled from 1 to 10, right? Pause the video, take 2 minutes and write down all your answers. Okay, I assume you have done that. So let's check what are the answers. The first organelle over here, this is nuclear membrane. This is nuclear membrane. And what is the second one? This one. This is nuclear pore. This is nuclear pore. The third organelle over here, this is nucleolus. This is nucleolus. Fourth organelle here, this is endoplasmic reticulum. This is endoplasmic reticulum. Fifth one here, this kind of structure, this is Golgi apparatus. Sixth one over here, this is mitochondria. Seventh one is a cytoskeleton structure. It is microtubule basically, right? What is the eighth? Eighth is a lysosomal structure. It's non-specific, but it's lysosome. And the ninth one is a vacuole. And the 10th one over here, this is a plasma membrane. This is your plasma membrane. Okay, nice. So just test yourself. What are your answers? So these are your answers. Please check. Okay, good. What are subcellular organelles? Cells contain various organized structures that are collectively called cell organelles, right? When a cell membrane is disrupted mechanically or using a lipid solvent like 2-20, the internal organelles, they are released and they are homogenized, right? So the organelles, they are separated by applying differential centrifugal forces. Albert Claude received Nobel Prize in 1974 for his work on the fractionation of cellular organisms. So what is this cell fractionation and why do we need to, identify all these separate organelles? See, cell fractionation, it's a process in which we separate the different organelles, right? This is achieved by centrifugation. So I'll explain you what I mean. See, this is a centrifuge. This is a centrifuge rotor. You can see this type of centrifuge is a hinged bucket containing tube, swinging bucket centrifuge. So what happens is that in this tube, it contains the solution which contains cell organelles, right? Initially, it is present in the vertical position, and then when centrifuge is switched on, these tubes are going to take this position horizontally due to the centrifugal force, and as a result of this force, the components are going to separate down. I hope you can appreciate here that the components are fractionated into two parts, right? Here it is a pallet and here it is supernatant, right? So basically the cells, they have been, the components have been separated. So this is the technique of cell centrifugation and how we use it for cell fractionation. See, this is the cell, and after using some mechanical methods or by using some solvents, the cell membrane is lysed. And the cell membrane is lysed, and all the cellular components, cell organelles, they come out into this solution over here. You can see there are multiple or the different kind of organelles suspended here. So we use this centrifugation force, and based on the gravity, that is the rotation, and for the time duration, we can separate various components. For example, if the centrifuge is run for 600 gravity for 10 minutes, first of all, nuclei in the nuclei settles down, right? And then when the speed is increased to 20,000 gravity for 30 minutes, these are the organelles which settle down. And if the speed is increased and for more duration, then other organelles are separate. So basically, this is how various organelles are separated on the basis of the amount of centrifugal force and the duration for which it is applied, right? So this is how we get different fractions of the cellular organelles. So coming to the concept of marker enzymes. What are marker enzymes? Certain enzymes are specifically located in particular cell organelles. So these specific enzymes, they are known as marker enzymes. So after centrifugation and separation of subcellular fractions, the presence of a marker enzyme help us to identify each organelle. Okay. So here in this column we have various organelles, and this is the time duration and the centrifugal force, and this is a marker enzyme. For example, if we're doing some experiment and if we find ATP synthase enzyme, so we can say that the organelle which is most likely to be present here is mitochondria, right? Similarly, if we find cathepsin, so we can say that cathepsin, so the organelle that is most likely to be there is lysosome. So these are the enzymes which are specific for the respective organelle. This is how we know which organelle we are dealing with. So we are starting with the different cell organelles, starting with nucleus. So we know what a nucleus is. Nucleus is the most prominent organelle of a cell, right? It is present in all the cells except mature RBCs. Mature RBCs do not have nucleus. Why? Because it's an evolutionary mechanism, so that the RBC has to carry oxygen. So it is giving more space, the absence of nucleus gives more space to the RBCs to carry more hemoglobin for oxygen carrying purposes, right? The uppermost skin cells may also lack a distinct nucleus. In some cells, example lymphocytes, spermatozoa, nucleus occupies most of the cell space. So looking at the structure of nucleus is surrounded by a double membrane, right? There are two membranes, inner membrane and outer membrane. You can appreciate here, this is the inner membrane. Let me change the color. You can see this is the inner membrane, this one, right? This one. This is the inner membrane, and the outer one is this one is the outer membrane, right? Which is this outer membrane is in continuation with the endoplasmic reticulum like this. So inner membrane is also known as perinuclear membrane, and it has numerous pores. And the outer membrane is in continuation with the endoplasmic reticulum, right?
Nucleus contains DNA, which is the chemical basis of genes which controls all the cellular functions. DNA complexes with proteins to form chromatin and is organized into chromosomes. So I hope you all know which are the proteins that is DNA form complexes with. These are histone proteins, right? DNA complexes with proteins to form chromatin that is organized into chromosomes. So what is the function of nucleus? It is site for DNA replication and RNA synthesis. Transcription. I hope you know what a central dogma is and what is transcription, what is RNA synthesis, right? Okay. Nucleolus appears as a lighter area within the nucleus, and it is site for RNA processing and ribosome synthesis. It is prominent in cells that is actively synthesizing proteins.